opportunity for innovation within the natural code of a seed is enormous, but has yet not been fully realized. We are now in a race against time. As humans, we only get one lifetime to make an impact. Of course, we need quality management. Of course, we need transparency on the employed technologies. Of course, we need to consider implications for farmers and breeders. However, we risk not realizing the potential of new breeding technologies if we are unable to align regulations and politics globally to ensure access to every nation. From a science perspective, there is no reason to believe that these technologies, if properly implemented, should be more risky than conventional breeding. To the contrary, they are more precise.
New techniques like CRISPR-Cas can achieve desired changes precisely at a targeted location in the genome. This can also be achieved in many cases by way of conventional breeding, but it would take substantially longer.
With regard to new genome editing, such as CRISPR, we see a number of risks that make a strict regulation of the techniques absolutely necessary. Some of them are specific risks associated with new genome editing. One example is the application of the technique itself that can cause unintended on- and off-target effects, which are specific to the technical process. For example, there might be an unintended insertion of DNA fragments into the target sequence of the target organism, or there might be effects outside of the desired target sequence. I would like to point out that the use of biotechnological mutagenes enables the circumvention of natural mechanisms of gene regulation and genome organization, and they also make the genome available for changes to a much greater extent than ever before. Genetic conditions, which could be hardly altered by conventional breeding, can now be genetically engineered.
The advantage of the novel genomic techniques such as CRISPR-Cas9 in plant breeding is that we can induce targeted mutations at previously defined target genes. This gives us the unprecedented opportunity to rapidly develop climate-resilient as well as resource-efficient crop varieties for the future.
Recent advances in gene editing technologies are generating very precise and efficient new tools to recover traditional crop varieties and produce new ones, to increase resilience and to produce healthier and more sustainable food. As an example, the worldwide cultivation of bananas provides economic and food sustenance for more than 400 million people. The crop is affected by the TR4 fungus, which destroys the plants. No fungicide can be found to control it. The application of CRISPR-Cas9 technology could be very encouraging. Crop plants should be evaluated and regulated according to what they are, not how they are produced. Plants obtained by mutagenic genomic editing, which does not introduce genes, should not be considered GMOs. These crops cannot be distinguished at the molecular level from others obtained by traditional breeding or similar spontaneous mutation, so the same rules must apply.
We need a differentiated assessment of new breeding methods, like genome editing, and a suitable legal framework that enables as many plant breeding companies as possible to use them. It is a positive step that the European Commission has acknowledged the potential of new breeding methods to ensure a sustainable agriculture and food supply.
The framework that is needed here therefore continues to include approval procedures with comprehensive risk assessment. In accordance with the precautionary principle, the technology-related risks must be assessed and the labeling and traceability of GMO products must be ensured.
These technologies can help unlock the full economic and environmental potential of seed. With clear policies and practices in place, we can collectively build a more sustainable future for agriculture in Europe and beyond.
There is no scientific foundation for the legal discrimination between mutations induced by genome editing or the mutations that occur naturally on the field.
That's why we stand up for changes in the regulations of all plant breeding techniques.
And that is also why we initiated the Give Genes a Chance campaign, which has been supported by around 500 scientists and early career researchers throughout the European Union so far.